very much to Professor Granovich and his team and Tatiana. It's been a real pleasure coming over. It's my first visit to Russia. It's my first visit to St. Petersburg, and I've been really well looked after, and you have a lovely city and a really great university, so thank you very, very much. So I'm going to talk to you today a little bit about the research I've been doing over the last 5 years on zooplankton behaviour in the Arctic. But I'd just like to start with a little bit of a background on myself. I'm a sort of benthic behavioural ecologist. I did my my undergraduate degree in biology down in Portsmouth, which is on the south coast of the UK. Then I went on to do a master's in fisheries biology in Bangor in North Wales, which is another marine institute. And then I went over to the east of the UK to Newcastle upon Tyne, where I did a PhD in polychaetes with Professor Peter Olive. And for a long time I've been really interested in biological clocks, and when I go to a biological clock conference, it's really great because I get to meet people that are working on all kinds of organisms, from Drosophila to humans to hamsters to mice, but very, very few of us actually work on marine organisms, and that's been my real underlying passion for my entire scientific career. And in the last few years I've been really fortunate to get back into marine chronobiology, which is the study of clocks and rhythms in the marine environment. So a little bit about SAMS, where I come from. This is a map of Scotland on the right, and for those of you who've been there before, you've got Glasgow, the bottom edge of Scotland. Scotland is not very big, but it's very underpopulated. So there's not many people. There's only 6 million people in all of Scotland, and most of those live in Edinburgh and Glasgow and Aberdeen on the east coast. And SAMS, which is the Scottish Association for Marine Sciences, is based in Oban, just there where it says "We are here," and it's a small research institute. There's about 150, 180 people that work there, and it specializes in marine sciences. And it's part of a university called the University of Highlands and Islands, which is a sort of virtual university which consists of a whole bunch of institutes, higher education and research in Scottish Highlands. We are generally considered the most academic and possibly research focused of the UHI. We have undergraduate students, so we have about 100 students, which is, for those of you who were here yesterday and I was talking to you, we think that's a lot. I think you've got something like 40,000 students here at the university, so maybe to you 100 students isn't a lot, but we started with only 3 undergraduate students. When I first started teaching at SAMS, I'd sometimes have 1 or 2 people in my class, which was bad for them because they couldn't sit in the back, and if I asked them questions they had to answer because there's so few of them. So the institute is based on a little peninsula that sticks out into a piece of water called the Firth of Lorne, which is this sort of great glen which runs up the length of Scotland, and some of you may have heard of the famous Loch Ness, which is just there, which is where there's a monster. Anyway, people think there might be a monster that lives in there, but we know it's not true. So the institute is based of teaching building here, and we've got a new sort of science park which is being developed, which is where our commercial team are based. The building is fairly new, 2004. It's fairly sophisticated, but the institute as a whole has been around for a long time. It was founded by Sir John Murray, who was a very eminent Scottish oceanographer. He went on an expedition in 1872, which was the Great Challenger expedition, which was a predominantly oceanographic expedition, and he was the first to describe the ocean trenches and measure the depth of the oceans. So really the great grandfather of physical ocean. Before he opened the Scottish Marine Station in Edinburgh, and it was a very prestigious place, based on a little boat, and then in 1897 it was relocated to a little island in the Clyde, which is on Glasgow on the west coast of Scotland, and that's where it actually stayed for quite a number of years, and then in 1967 it was actually moved to the current location where we are now. So although it's quite a new looking building, and it's not very big, it's actually it was established a long time ago. So on this great journey of historical interest, we flash forward to some other things which might be of interest to you if you come to Scotland and Oban. Oban has a very famous distillery called the Oban Distillery, which makes a very famous and very nice Oban whisky. And Scotland, as you may know, is famous for different whiskies. So if that's what interests you, then that's great. The other thing is we've got lots of nice wildlife. We've got these things called Highland cows, and tourists really like this. So they stop in the middle of the street in their cars and they look at the cows and take photographs, and they are very impressive. They've got these great big horns, and they're sort of red and shaggy. So I arrived at SAMS in 2007. I got a research fellowship there, and I thought, when I arrived, oh, this is absolutely great. This is a lovely place. I can go kite surfing. I can go for walks. The water is blue and clear. I can be a marine scientist and enjoy myself every day because it's just wonderful. Unfortunately, it's not always like that. Most of the time it's actually like this, and actually it's not very nice at all. It's one of the most beautiful places on the planet, I think, when the sun is shining, but when it isn't, it's probably one of the most miserable places. It's certainly raining a lot of the time, and it's dark in the winter months. However, it's not as dark as in the Arctic Circle during the polar night. This is a cartoon which was drawn for one of Jürgen Berger's papers, and it shows the concentric rings of twilight that you get during mid-winter. So we are about here, and this is silver twilight, and this is nautical twilight, and this is astronomical twilight. So for 6 months of the year it's dark here, and for 6 months it's continuously light. So the North Pole, you get 1 day and 1 night per year. And then you get these various shades of twilight. In the civil zone, the sun at midday only reaches to be up to 6 degrees below the horizon, so it's always below the horizon. Nautical, it's 12 degrees, and astronomical is below 12 degrees. So even at midday, the sun doesn't come any higher than 12 degrees below the horizon. So it truly is very, very dark. And my feeling about living in Oban was actually changed by this man Jürgen Berger, because I realized that although I was living somewhere that I thought might be a bit horrible and dark somewhere sometimes, it certainly isn't as dark as where Jürgen Berger lives, which at the time was at Unis on Svalbard. So that made me feel a little bit better. And actually after that I spent most of my winters studying Arctic biology up around Svalbard, which is great because in the winter everybody thinks you should go somewhere tropical and nice and warm, but then you come back and you feel miserable about having been somewhere nice and warm. So what you should really do is go somewhere really, really cold and dark for your holidays or work, and then you come back and you think, well, it's not that bad after all. So Jürgen Berger came to SAMS on a sabbatical. Jürgen Berger is a professor now at the University of Tromsø. His training was in taxonomy, but he became really interested in polar night processes and how marine organisms would respond during the polar night. And he came to SAMS on a sabbatical to write papers and to work with an oceanographer and a good friend of mine, Finlo Cottier. And he gave a lunchtime seminar one day, actually after he'd been there for about a week, and he presented these data. And I had just started my job. I had been working at Newcastle University before that. I'd spent all of my time looking at patterns in data, and most of my data was quite sparse, so there wasn't very much of it. I had to do a lot of work to collect enough data to do my analysis on. So when I saw these ADCP data, which are spanning many, many months with lots of patterns inside the data, I thought, this is great. I could do some chronobiological analysis on these data and see what sort of patterns and rhythms we've got in there. So I said to Jürgen, I said, well, why don't you give me the data and I'll just see what we get. And Jürgen said, absolutely, that's a fantastic idea. He's very, very charismatic, and he likes to share information. And he said to me, go ahead, Kim, look at the data. So we did. It's just showing you what an acoustic Doppler current profiler is. It's active acoustics. It's an instrument you put onto the seabed. It's used in oceanography to measure ocean current speed by sending out an acoustic signal. This particular one is at 300 kilohertz, and then that signal bounces off particles in the water, whether they be inorganic or organic particles like zooplankton, and then it measures the Doppler effect, the time it takes for those particles to come back, so you can tell how fast those currents are moving. It's the workhorse of oceanographers. That's how they measure the flow of currents, and you've got lots and lots of these all over the oceans. Okay, So what we were seeing, what was in the acoustics was down to biology, and we were seeing the effects of diel vertical migration. Because of the frequency of these acoustic Doppler devices, we knew that they were in the zooplankton range. There may be a few small fish in there, but generally the majority of the patterns we were seeing was from zooplankton. And I know that those of you who study zooplankton will say, okay, well, there's lots of different types of zooplankton. Of course there are. So it's very much a population response that we were seeing. For those of you who don't know about diel vertical migrations, basically the zooplankton come up at night and they feed on phytoplankton and they go down during the day, and the response is cued by daylight. It's one of the largest migrations on the planet. It occurs every single day. You get lots of freshwater zooplankton that do the same thing, and the general pattern is for them to move up, to feed, to sink down, to defecate at depth. And that's really important because they're bringing carbon down into the deeper ocean, and then they swim up and then they feed and then they go down, and the process is repeated over and over again, and it's driven by light. So my question was, well, what would happen during the polar night? And so we had to look at these data. So generally there was lots of it, so it would span many months, and if you draw a graph that long, it would be fairly useless. So we used something which is called a double plotted actogram. So these data, this is an example of what we do. This is sort of day-night data, and then we cut it up. And then we stack the data on top of itself, like so. So that's a day. And so you just stack the days on top of themselves, and it's a really good way of visualizing rhythmic behavior. And I've just got an example here: circadian rhythms in flies. This is Drosophila on your left, and a hamster on your right. They're both studied a lot in chronobiology. Hamsters like to run in wheels, and you can measure the wheels going round, so you can measure their rhythmic behavior. For any of you who've kept hamsters, some of you will have kept hamsters. They're really annoying because they're only active at night. So if they're in your bedroom, then they make a big noise. So it's annoying, isn't it? It's a pain. I don't know why anybody's got hamsters. Guinea pigs are much better. We've just got guinea pigs. They're active during the day. Anyway, you get these double plotted actograms. And you can see here, this is day, night, and they're active at night, and then you turn the lights out and something changes. They're still active, but the activity is earlier every single day. And that's because the period of their circadian rhythm is slightly shorter than 24 hours. So it gives you a really quick, easy way to visualize the data. So we really wanted to know what was happening during the polar night. So we went out there, we had our acoustic instruments, and we looked for patterns. And the paper that started all this in 2009 had this sort of key figure from one acoustic mooring in Kongsfjorden in Svalbard. Let me just sort of walk you through this. This is all the way from October through to March. Up here, this is when the sun is still above the horizon, but there's less than 6 hours of daylight a day. This is when the sun is permanently below the horizon, and this is when the sun is again above the horizon, but only for a short period. And what we did was we used autocorrelation analysis, which basically gives you a strength of the rhythmicity, a strength of the migration. So if there's no migration at all, then this number is very low. If there's a really nice pattern of migration, then the autocorrelation provides a very high number. So the darker the box, the more rhythmic you have the migration. And this is what it looked like. This is the pattern. So clearly towards the autumn and the spring, it was more dark, so there's more rhythmic migration. But then in the middle, even in the middle of winter, in December, January, you still had vertical migration, but it was restricted to this sort of depth, which is really interesting, because you'd think that any light that is there should be at the top. So why isn't the migration up here? And that's what we then subsequently found out the reason for. So we published this, and it took a while. One of the biggest criticisms from referee number 3. It's always referee number 3, isn't it? The first two are great, and you go, oh yeah, got it, got the paper in, yes. And then you read referee number 3, and you go, oh. Another year, another project student, another PhD student. So referee said, yeah, this is okay, this is fine, but it's only one fjord in Norway, okay? How representative are these data of the entire Arctic Ocean? I'm not quite paraphrasing, but something along those lines. So we knew deep down that referee number 3 was a bit of something that I can't say in public, and certainly not on YouTube. But the message was, go figure that, smarty pants. You know, if you think you're so clever, go figure that. And we thought, okay, right, let's see what we can do. And fortunately, my good colleague and friend, Photoperiodism, has a lot of friends in the oceanography world. So he and I wrote a little proposal, and we got a little bit of money, and all that involved was for us to contact all these people, many in the States, Washington, Canada, Norwegian colleagues, to give us their ADCP data. All 300 kilohertz, all the same from all their moorings, and they had around about 40 data sets that they gave us, collectively spanning something like 50 years of data. So a vast archive, and we called it the Pan Archive. And Estelle Dumont, who's our technician, robotics technician at SAMS, she's really good on MATLAB, and she put it all together in a fantastic database. So then we could interrogate that database, and we thought, okay, Pan-Arctic, this is what we want to learn about. And this really was the point at which Laura Hobbs, Finlow and my PhD student, came on board, and we generated actograms, and that's what these are. I've just shown you one, and we generated hundreds and hundreds of actograms. We spent 2 or 3 days one day in a lab where the floor was clear, and we just put out all these pieces of paper on the floor, and we were trying to put the ones that looked similar together, and we were trying to look at patterns. It was almost like a work of art. But what you can see here, this is from one station, happens to be Kongsfjorden of 1 year, is you've got 3 different actograms from 27 meters, 51 meters, and 103 meters. And think of these as slices through the water column. And what you can see here is you can see diel vertical migration. This is the middle of the day here and there. Okay? So, and this is the middle of the night. So there's a lot of backscatter in the middle of the night and very little backscatter in the middle of the day. And if you stack every one of these is one day, one strip is one day and then another day, what you can see is that there's a lot of activity at night, and the onset and offset of these lines correspond with day and night. So as the days are getting shorter and shorter and shorter, so the amount of activity during the day is getting less and less and less, and the amount of activity at night is getting more and more and more. And then you can see how this gets less and less the deeper you go. So the depth and the time of the maximum amount of response actually changes with depth. So that was really nice. But what we noticed was patterns in the data. So what you can see here is these lines going across here like that. So here you've got not much backscatter, so there's not much at 27 meters, and there's quite a lot at 50 meters. And the same here, there's nothing at 27 meters, and there's loads at 51 meters. So red is a lot of backscatter. That's where the organisms are. And what we realized, this is just showing you the solar altitude. This is Sivir twilight and this is Nautica twilight. Apologies, these lines should be smooth. They shouldn't be like that. What we realized was that this is related to moon phase. So at the beginning we didn't really know how to deal with lunar data. We didn't know how to quantify it in terms of output, because there's the brightness of the moon, but there's also the height of the moon. So we've just plotted everything for the sake of it. So this is lunar altitude. The lighter it is, the higher the moon is. This is lunar illumination in terms of the phase of the moon, and then this is a sort of average that we came up with between these two. Either way, what you should be able to see is that when you've got a very bright blob here, you've got little backscatter there. So there was clearly an effect of moonlight on zooplankton migrations, at least down to 50 meters. The story is a little bit more complicated because you can see there's another column for cloud, and that is an extra factor which actually influences that, but I'm not going to talk about that just now. So what we did was we put all this together from the entire archive, and we quantified it, and we characterized two new types of behavior. So if you look at the whole month, okay, and you get these blobs here and here and here, the distance between these is the distance between full moons. So it's a lunar month response. When the moon comes up and it's bright, the zooplankton go down. But what was really interesting for me was that at the same time, during any one of these, what you would get is you would get another rhythm of activity, which was about 25 hours long, and 24.8 hours to be exact is one lunar day. So that's the time it takes for the moon to come up and set, and every day it's slightly later, of course, so you go up and down, and so every day the period of the moon at the same point in the sky is 24.5 hours. So what we were noticing was that not only did the zooplankton respond to the moon coming up and down, but they also followed the moon as it went up and down above the horizon. And I've just got a little summary here with all the data. So you've got 4 full moons over the winter period, from November, December, January, February. Okay, and the ones closest to the edge are very close to 24 hours, whereas the 2 full moons in the middle of winter, around the winter solstice, there's a lot of variability, admittedly, but they are shifted to a longer period, to a near 25-hour period, which is a lunar day. So just to summarize, a lunar day is the moon coming up and down above and below the horizon, a bit like the sun does, and that's 24.8 hours long, whereas once a month you get the moon coming up and being up in the sky all night long, and then the zooplankton migrate down. So we published that paper, and we sent it to Current Biology, and they accepted it, which was fantastic. And then they said, well, how about doing a video abstract for the paper? And I'd never heard of a video, what that was, and they said, well, basically you need to make a video with your phone or something, or a GoPro, that describes your science in about 6-7 minutes and makes it really exciting. And so we have a resident filmmaker who works at SAMS, and I said, look, Andy, we're going out on a cruise up Loch Etive, which is this sea loch next to SAMS. We're actually collecting zooplankton. Shall we just try this? And we were out for 2 days, and so we made this little movie, and it was a lot of fun, and it caused a lot of interest. And so I'd just like to show it to you. It's about 7 minutes long, and it sort of summarizes what I've been talking about. So let's see if it works. Down with the rising and setting of the sun. We're here in the middle of winter, and it's cold and fairly dark. There's not much sunlight. It's a good time to see these migrations happening. So why do the zooplankton undergo these daily migrations, billions of them moving up and down in the water column? Well, the reason is actually quite simple. They move away from the sunlight because there are predators in the water. The zooplankton themselves are composed of sheep and tigers. There are zooplankton that are grazers. They feed on microscopic algae and plants. And there are other zooplankton which are like tigers and now feed on those grazers. Small fish will hunt the zooplankton, and out at sea large whales will eat the zooplankton. They're the basis of the food chain, and they're incredibly
important. They're all visual feeders. They need daylight to feed. And that's why as the sun comes up, the zooplankton migrate down into the ocean depths to get away from that predation. And our question was, well, what happens in environments where you have no day-night cycle? This may sound unusual to some, but if you go up into the Arctic, for 6 months or so, the sun sinks below the horizon. There's no daylight, no solar daylight to speak of. And what happens to these migrations? Do they still exist? Are these prey still being hunted by predators? And our findings are that they are, but the story is actually quite different than what we expected.
So here we are in the ship's lab. We've just been outside, brought the nets in, and now we're sorting out the zooplankton. So what was this finding that we discovered in the data collected in the Arctic about zooplankton migrations? Well, our approach was to gather data, to mine data from a large international group of scientists who have moorings up in the Arctic. Moorings on the seabed with acoustic instruments on them. And the acoustic instruments allow us to visualize zooplankton migrations. The data that we used was massive, 20 years worth of data, cumulatively spanning 50 years. And when we mined this data, we found something which was hidden. And this was very exciting. There seemed to be a coincidence of migrations with moon phase. We discovered that the zooplankton respond to moonlight, and this was something that nobody had really expected. So every time the moon comes above the horizon, zooplankton will migrate down to depth, and then as the moon sinks back below the horizon, they will come up again, a little bit like what we have here in Loch Etive when the sun comes and goes down. But the lunar cycle, the lunar day cycle is different. It's 25 hours, it's not 24 hours. So this is a completely new finding and one that we hadn't expected. But what was even more exciting was that once the full moon had got high enough and bright enough in the sky, every month for about 6 days, there would be a massive migration of zooplankton down to about 50 meters. And we now think that the reason why this happens is because there's probably werewolves in these waters. There are predators that feed on the zooplankton like Thermisto, and they will actually actively hunt the prey, these copepods, the grazers, down to depth. And we know that they can do this because we have measured their visual spectral sensitivity, so we know that these animals can detect moonlight. And moonlight provides enough light for these werewolves of the deep to actually be out there hunting. And the consequences of this are manifold. We know that zooplankton migrations move carbon from the surface waters into the deep, so they're very important for global carbon cycling. And we also know that zooplankton aggregations structure the patterns and migrations of other predators, such as polar cod and whales. But perhaps most important is that we now have a completely different perception of the Arctic and the polar night. That there's a baseline, there's a different baseline. We'd expected there to be nothing happening at this time of year, when it's incredibly cold and totally dark for months on end. And yet we now know that the moon drives these migrations probably because of predators, which is incredibly important about making predictions for the future, where the Arctic is changing
and where we have ice retreat. And effectively, we will have more light penetration into the oceans. So werewolf migrations may become more dominant in the future. Werewolf story, and then we ran with it, and we actually put werewolves into the paper. We sort of mentioned werewolf, and the response was huge. The public outreach was enormous. Everybody was talking about it. We even had. I was contacted by a group which have a website called the Werewolf Appreciation Society. Okay, and they wanted to interview me about zooplankton migrations. And I was thinking, am I a serious scientist here, or is this getting a bit silly? But my point is that people were talking about zooplankton that would have never talked about zooplankton before. So, in a way, we were using different forms of media and communication, and it just so happens that we did the right thing at the right time. And it was a fun time, but in all seriousness, it also, we felt that we'd done a better job at communicating our science to different audiences. Okay, so you may ask, so what, what's the big deal? And this is really something that we've been working on ever since. As I mentioned in the movie, there's carbon associated with geovertical migrations, and there have been a number of papers published on this. So we wanted to quantify this. And so we put down a sediment trap in Isfjorden over a full moon and a new moon period. So this is a acoustic backscatter in Isfjorden over the new moon period. You've got depth down the side here, and you've got local time. So this is the beginning of December in Isfjorden. And what you can still see in the middle of the day is you've got this little bit of a migration downwards. You can still see some geovertical migration here happening. Compare that to the full moon. It's quite a different picture. Unfortunately, we can't look at the close surface with these acoustics, but you can see that there's a lot less, it's a lot bluer up at the top. So there's very little back scatter here. So the majority of the animals are between 30 and sort of 60 to 70 meters. But they've clearly moved away. So we just happened to, well, not just happened, but we placed some sediment traps at what was it exactly? I think we were 30 meters and at 90 meters. So we had two sediment traps there. And what we really wanted to do was we wanted to catch the swimmers, and we wanted to quantify the migration. We wanted to determine who was doing the migrating at what time. So the sediment traps were opening and closing every 6.4 hours, and we were trying to determine a geovertical migration within the zooplankton community. But unfortunately, that was that was lost in the noise of the data. So instead, we focused on fecal pellets, and the results were quite interesting. So first of all, this is the new moon period, and this is the full moon period. What you've got here is you've got the top top sediment trap at 30 meters, and here you've got the one at 95 meters. And the first thing that you notice in terms of fecal pellets is that there's a lot more down in the bottom sediment trap, which is not surprising. It's what you'd expect because you're catching a lot more fecal pellets from the animals above. But then just comparing the two bottom ones, we're seeing approximately 30% more fecal pellets in these, and hence when we calculate the corresponding carbon flux, we've got a lot more carbon flux during the full moon. So our hypothesis is that during the full moon there is a migration down. The animals are constrained in a smaller space. Quite why they should possibly move up, we don't know. But that there's a lot more predator-prey interaction happening. And actually, the euphorsids, Physanoessa, etc., are probably feeding on copepods. And so that is a consequence of these lunar migrations, or at least that's what we're proposing. A lot more work is still required on that. So actually, it is a bit horrible, but it's supposed to show you that it's not vegetarian fecal pellet matter. It's actually carnivorous fecal pellet material. So another thing that you can do, and this is what Laura did as part of her PhD, she used the whole pan-archive data set to quantify whether or not DVM is continuous, so you can see it happening all through the year, or it's seasonal, so that it stops for part of the year. So here we've got DVM, and here we've got DVM again in the autumn and the spring, but in the winter months it stops, or there's nothing at all. And you can see that the latitudes that these acoustic instruments were based at. So taking the whole pan-archive with various caveats and constraints, you can sort of plot this, that in shallower waters the onset and offset of DVM is constrained, whereas in deeper waters the distance between the onset and offset is actually much greater. What's really interesting about this is that the dates of DVM stop and start over the whole acoustic archive accurately predict the date of the winter solstice, the middle of the winter, by just a couple of days. And you can see the error bars on this. I mean, isn't that just phenomenal? That the population response is actually centered around the winter solstice so accurately. So they clearly are really, really responding to the light-dark cycle in a very dramatic way. And if you classify the whole pan-archive data set between continuous DVM, seasonal cessation of DVM, so when it stops during the winter, and no DVM, you can see that the clear circles where it's continuous, and here it's seasonal, and then here you've got no DVM, are defined by latitude. And actually there's very little relationship with sea ice cover. So sea ice cover doesn't seem to make a big difference to DVM, other than at the North Pole we've got no DVM at all. Unfortunately, we don't have any data for these latitudes yet. So I don't know if you wanted to stop for a break. I think we're about halfway through, and if you want to stop, or I just carry on.
Let's have a short break.
Yeah.
Usually it brings in some discussion.
Because I'm back to quite set.
Okay, so I think at this stage I thought that I'd done enough work and I would have a summer vacation, and my idea of a summer vacation is to go kite surfing in Oban. That's what I did, but unfortunately I discovered a very hard place which is just there, and the result was that I hurt my foot and I was on crutches. I actually didn't break anything. I cut one of the tendons in my foot, so I couldn't move my toe. But the consequence was that I had to be in crutches or in plaster and in crutches for 6 to 8 weeks. So I wasn't allowed to go to work, which was sort of good, but it was also a little bit annoying, until I realized that I should take advantage of this, because just at that time NERC, which is our main funding agency, the people that give us money as scientists, as natural scientists, they'd put out a call to fund changing Arctic Ocean work, and a couple of descriptors there which really met what we were interested in was a description of changing seasonality, so that's day-night cycle, and its subsequent impact on biological processes and ecosystems. Yes, that's a really good thing to see that in a funding call. Sensitivities of multiple stresses and development of models of the life cycle of key species. We clearly had to work with ecological modelers to be able to answer some of the questions that we were now posing. So I spent my 8 weeks lying on the sofa at home with my laptop. I couldn't do anything else, writing a grant. And I tell you, that's the best thing to do. If you want to write a grant or a paper, hurt yourself, break a leg, not an arm or anything like that, or anything really serious, but just make yourself stay in one place. You can't move, you can't do anything, and you will write a good grant. So it allowed me to have a good think about what we wanted to do. We all know that the Arctic is changing. This is a model projection from NOAA showing temperature anomalies between 2010 and 11. So the really red stuff is way above the annual mean. And so there's a lot of data out there now showing how the Arctic is warming. And we also know that animals like zooplankton, these copepods, the species you get in the Arctic, are generally changing distributions and habitats as a consequence of these warming waters. And a number of modelers have made predictions. There's a group at Strathclyde who are sort of leading the modelers under the NERC Changing Arctic Ocean program, and their collaborators. They've published a lot of data showing that over time, certainly Calanus finmarchicus, which is one of the dominant North Atlantic zooplankton species, is heading northwards. However, as I showed you before, the Arctic day lengths are not changing, they're remaining constant. So the photoperiod is remaining constant. So what we've got is we've got animals which are moving from an environment, and they're tracking isotherms they're moving northwards into a different photoperiodic environment. And so what we've got is we've got a thermal photoperiodic mismatch. These animals have previously lived in an environment where the day lengths over the year were of a certain flavour. Now they've moved into the twilight zone for at least half of the year, and they're going to get different photoperiods. So what? Well, some people like Stein-Cartwright have published papers saying that things like mesopelagic fish, the reason why you don't get many in the Arctic, you get them all over the oceans of the world, but you don't get many in the Arctic, is because of photoperiod. There's not enough light for them to feed, etc., etc. That's a constraining effect on the migration of these organisms into the Arctic. Why else should mesopelagic fish not be common in the Arctic? And perhaps you could argue, well, maybe he's wrong, maybe it's not quite right, maybe there's another reason. But anyway, it's a hypothesis, and it's a fairly reasonable hypothesis. So we can quote, as marine organisms track isotherms, there will be trade-offs between latitudes of favourable temperature and potentially unfavourable day lengths. Okay? Now, you could ask, well, so what? Well, if you are a bird, if you are a quail or sparrow, and you are a male, you will certainly physiologically at least know about photoperiod, because male birds, many male birds, especially these two, have got testicles which appear and disappear depending on the photoperiod. They don't need their sex organs at times of the year when they're not needed, so they grow them when the days become longer. Okay, very important. Why have extra baggage if you're flying around? There's no need for that. And in terms of the insect world, okay, we know that with increasing latitude, the critical photoperiod to induce a sort of a physiological condition, normally a reproductive condition, increases with latitude. So the further north you go, the longer day lengths you need to induce something to happen. Photoperiodism, the response to day length, is orchestrated through the circadian clock. And the circadian clock is an autoregulatory negative feedback molecular machine, and I'll talk a little bit more about that later. But the point is that you've got a biological clock that's ticking away, that tells you how long a day is. If you've got different amounts of daylight or darkness in a day, then you need a reference point, and that reference point is the circadian clock. So circadian clockwork is involved in photoperiodism. So I'm building the story up here. And we know that from the insect world, so Bradshaw and Holzapfel published this in a pitcher plant mosquito, but there's many other examples that if you mix up the thermal year, so the temperature cycle year, and you mix it up with the day-night cycle year, you will get changes in fitness. Okay, so we all know that at a certain time of year you have got certain plants that come out of the ground, crocuses say, these little beautiful flowers that come up. They may be induced to flower because of day length and or temperature, but they will have set critical thresholds, and if you change those thresholds, if you mix up the temperature and the photoperiodic signal, then often there's a real consequence to that. Okay, so I used all that in my proposal, and I said, look, this is really important, nobody else has thought about it before, give me the money. And they did. So I got funding, which was really nice, and I got funding with a good colleague of mine, Bettina Meyer from the Alfred Wegener Institute in Bremerhaven, and also affiliated with Oldenburg. She's a molecular biologist. She's interested in circadian clock genes. So approach that we developed for studying Arctic zooplankton, we would use her expertise in molecular clock genes, and we would see if we could decipher what was going on. So why copepods and krill? Maybe I don't need to tell you this, you'll know it anyway. They're the base of the food chain, very short food chain in the Arctic. You've got zooplankton, then you've got some polar cod, a few other species of fish, but not many. Then you've got seals and polar bears, and that's it, sort of. It's not very long, whereas in Antarctica it's a much longer food chain. So they're important in terms of the food chain. Copepods, Calanus finmarchicus, glacialis, hyperboreus, they've got these tremendous lipid reserves. They're great big fat things that gobble up loads of phytoplankton during the spring bloom and the summer bloom. They produce lots of lipids, fatty acids, wax esters, which are terribly useful for all the other higher trophic levels. Krill are important as well. They can be super abundant. They're not as dominant in the food chain in the Arctic as they are in the Antarctic, but they are still very important. Very important for carbon sequestration. So diel vertical migrations have been shown to contribute tremendously to carbon transport into the deep sea. Calculations are based from about 15% to 50% of normal gravitational flux of carbon. So diel vertical migration is very, very important in terms of carbon sequestration. It's also important in terms of the lipid pump, which is these organisms go into diapause, they're full of lipids, they respire at depth, so they're taking carbon down there, and they're not taking nutrients down, so they're not depleting the surface waters of nutrients, and that's a process which is known as the lipid shunt. The role of light in photoperiod, well, I've already demonstrated that. We know from colleagues, John Cohen at Delaware published a visual sensitivity. They're highly sensitive to light, both krill and copepods. Anna Batnes, who was one of Jürgen Bergert's PhD students, did some really nice experiments on day length. Laura Hobbs has published this pan-archive work, and we know the influence of the moon is very important. And Bettina Meyer's lab at the Alfred Wegener Institute has published work on krill circadian clocks, and one of her PhD students, Søren Hafka, showed that there's active clockwork in Calanus finmarchicus. So good organisms to work on. So we had an overarching hypothesis, which is that circadian clock rhythmicity, which determines behaviour and physiological responses in co-opted by a changing environment, altering Arctic ecosystems' functions. That's very, very long-winded and possibly a bit dull, but you need an overarching hypothesis in a big project. So this is our approach. We've been using something which is used in the Drosophila world to monitor Drosophila activity. So we've got our wild population of zooplankton. We go out with a net, we sample these animals at different times, we take the copepods up, we put each copepod into a 2.5 ml test tube, and we put that test tube into this rack with lots of other individual copepods. So we've got individual animals. and it's identical to Drosophila. We're treating Calanus phenmarchicus like Drosophila. It's the same thing, except that the tubes are vertical, not horizontal. If they were horizontal, the water would flow out, so that's not a good way of doing it. Then we get activity cycles. So this is day, this is the time, and this is activity, and we've got these different phenotypes. We've got some nocturnal ones and some diurnal ones, all sorts of interesting individual behaviour. So copepods are all individuals. They do their own thing. They seem to be very different. Some of them do one thing, some of them do another thing. We have no idea yet why. And we can sample these animals, we can pick them out, and we can do seasonal transcriptome work and look for clock cycling. We can also take our individuals here, and we can do individual respiration experiments now. So modern respirometry, you've got these well plates, individual well plates, about 500 microliters. You can put one animal in there, and there's a carbon fiber optode which is attached to the bottom of the well, and you can measure the respiration rate of one individual copepod. So we've got the behaviour of an individual copepod, we've got the respiration rate of an individual copepod, and then of course we can do all the classical stuff that we all love to do, and that's we can measure the length and the width and the lipid sac, and we can sex them and stage them. And with all these data then together, we can then talk to sort of the modelers who will determine optimal DVM patterns and understand any fitness consequences, which may then help to explain the sort of patterns that we get in our acoustics. So it's a sort of really a broad environmental genomics approach. And it may look sort of quite complicated, but at least, you know, this bit to that bit to that and that only takes about a week. Well, this doesn't take a week, and that doesn't take a week. That takes years, months at least, and this takes a long time too. But all this data is actually collected reasonably quickly. So we've been on 5 Arctic cruises now recently, and we've used this approach, and we've behaviourally screened 3,161 individual animals, and we've screened about 9,960 individual respiration incubations. So we have an enormous data set, and we need a whole team of people to actually analyse this, which we haven't got. But anyway, it's nice to have. So I'm just going to show you a couple of slides, some very preliminary data. We've been churning through this. We've been doing a lot of work around Svalbard and the Barents Sea with another project led by Phenocotier on the changing Arctic Ocean called Arctic Prize, and with Jürgen Berger on his project Arctic ABC. And so we've had fantastic sampling opportunities in the fjords and on the shelf and on the slope, and now as well in the fjords near Tromsø. And we focused first of all on just winter activity, polar night activity. And if you look at these stations, the outliers here on the right are all fjords. And so what we're finding over and over again is that swimming activity during January is much higher in the fjords. So the fjords seem to be much more productive. There seems to be more going on. So certainly in terms of just simply swimming behaviour, there's much more activity in the fjords. With the behavioural assay is that we discover all kinds of things that we didn't expect. So looking at just one data set from Ripfjorden in the northeast of Svalbard. We looked at adult female Calanus glacialis, and this is time along the bottom over 1.5 days, and activity at the side. And what you can see is that these animals are really quite inactive. However, take adult male Calanus glacialis, and they're really, really active. Not only are they really, really active, but their respiration rate is 2.2-fold that of the females. So they're really going for it. And the reason why they're really going for it is because they are in love. They're looking for the females. I think it's quite appropriate on today to talk about this. They're looking for the females, and they are following pheromone trails. And so when we take them out of the sea and we put them in our tubes, we still see this behaviour. And when we were doing these experiments on the cruise, there were lots of people that were saying, what happens if you put a male and a female together in the same tube? Or what happens if you put two males in the same tube? And everybody got very silly, and we had to do these very silly experiments. There's a dramatic difference in season. Here we've got a trace of adult female, about 32 animals here, Calanus finmarchicus, adult females in January, and compare that to May. So this is, just after the spring bloom, a tremendous amount of increased foraging activity in these animals. Okay. About copepods, what about krill? We've done a fair bit of work on Physanoessa enormis. John Cohen, who's a physiological electrophysiologist, he's really interested in visual sensitivity and bioluminescence, and has some really fantastic kit for determining visual sensitivity in these organisms that have very large eyes. So what he did was he measured mass retinal responses to these flashes of light, very short flashes of light. So the actual electrical activity within the eye. So you put you actually pierce the eye, and you measure the electrical activity to flashes of light. And the other way to do it is to leave them continuously and give them flashes of light over long time periods to look for any rhythms in visual sensitivity. And the results were really, really quite stark and very interesting. So irradiance is another word for light intensity. So this is much brighter, and this is much darker, and this is just the response in terms of the electrical voltage output. And so what you've got here is you've got a day and a night. So the day is in light and the night is in dark, and so it's this curve with undersaturation here and oversaturation there. So with increasing light intensity, you get an increased electrical response in the retina. But the point is that the animals collected at night and animals collected during the day showed different visual sensitivity. And these had actually been maintained in the lab for 24 hours before these experiments were done. So the visual sensitivity difference was endogenous. It was part of the circadian output. And it shows that they are visually more sensitive at night than during the day. And the amazing thing is with this, we were at 81 degrees north at the beginning of January, and it is absolutely dark, and these animals were caught at 50 to 100 meters, and still they have this cycle of visual sensitivity. When we kept them under constant conditions, and admittedly this is only data from one animal, although these experiments are hugely involved, so you don't get many replicates. You've got this peak here and you've got the peak there, which coincide with the darkest period of the solar day. And what you can see is even at midday here, the sun is only 12 degrees below the horizon. So there seems to be an endogenous cycle of visual sensitivity. Okay, so if you want to know about the circadian clock, what do you do? Well, you work with clock biologists, and this is it's a negative feedback loop, and you've got a gene here, and this is the period gene, and that expresses a protein, the PER protein, and that feeds back on its own transcription and turns it off. And once it's completely off, it starts again, and this process takes 24 hours. Okay? And I've got a lot of detail about that, and I'm not going to go through that now because it's a bit too late. Where do you do this kind of work? Well, you can do it up in the Arctic, but it's much easier to do it in Scotland because we've got a captive population of Calanus finmarchicus up here in Loch Etive, which is right next to the lab. So this is the lab, and we take the little boat and we go up here, all the way up there, and all the way around the corner here, and then about there's a big deep hole. It's 140 meters deep, and we've got lots of Calanus finmarchicus there. And we know that they have got that they show DVM because we have an acoustic instrument in the water. And for this particular project, we had two fantastic students, German students. Lukas Hooper, who was only doing his undergraduate degree, and Søren Hafke on the right, who was doing his PhD. They came out every couple of months to Oban, and we went out on the boat and we sampled copepods every 4 hours for 1.5 days, day and night. And the video that I showed you showed actually that's what we were doing at the time. We did all sorts of experiments here. This is a vertical migration column. These are lots of cameras here, got infrared light sources, and we were filming the copepods going up and down in this tube. And if you get the whole population, you can see these kind of rhythms of swimming behaviour. Now, you may not be convinced by that because it's a bit noisy, but we now know that actually, if you look at the individual swimming behaviour, they do it really rather well. This is using the equipment we've got now. This is a nocturnal animal swimming every night here, just to demonstrate that in case your mind switched off. That's what it looks like. And if you missed the last bit, that's really important. I'll play that again in slow motion. So there we've got an individual animal going up and down under constant conditions. And this peak here is the holy grail of this data set, because this shows that it is an endogenous circadian clock. It's doing something spontaneously without any day-night signal. It's actually swimming when it thinks it should be swimming, not when it's got the environmental information that it should swim. And Søren pulled out a whole bunch of clock genes. This is clock and timeless, clock and Tim, and what you can see here is beautiful cycling under the day regime, and then it carries on even when the lights are continuously turned out. So that's a real nice demonstration of circadian clock expression. Here's another one, per1 and clockwork orange. It's another core circadian clock gene which is cycling really rather nicely. And what was interesting, this would change over the seasons. So in May we'd get really, really nicely cycling clock genes, but by June these animals had gone into diapause, and actually we weren't seeing that, except in the following year the rhythms were actually in antiphase. So there's still quite a lot of work to be done there. Another cycle with clockwork orange, but generally the take-home message was that the rhythms were weaker with greater depth, but there was a seasonal component involved in these cycles. And Cyril pulled out all kinds of genes associated with metabolism, with feeding and lipid accumulation, and all kinds of things. And this story of a kind of seasonal change in the whole metabolism and phenology of these organisms is very, very apparent, and this is really what we're trying to now get more of a handle on, to really begin to understand that. Okay, so I've just got another 2 or 3 slides, and this is all a bit of fun. When I was doing my PhD, I went on a train ride and I picked up a paper that was written by a guy called Krasinski, and as you're well aware, Krasinski is a Russian name, and Krasinski wrote a paper called Dynamical History of the Earth-Moon System in Celestial Mechanics and Dynamical Astronomy, and he was based here in St. Petersburg. Okay, so he hasn't published anything much yet since then, but he was part of the Russian Academy of Sciences. And I picked up this paper, and it was like it was amazing. It was absolutely amazing, because he said, or he calculated, that the Earth has been slowing down. So this is empirical evidence from the fossil record. But actually, and this is the current day, and this is going back in time, so 600 million years, 1 billion years, 1.8 billion years, and then the Earth was formed around here. And at the moment, the day length is 24 hours, but here at the sort of Cambrian time, it was only 23 hours, and then going back was less and less and less, and then there's something that he couldn't explain here, but that might be quite important. But the take-home message for me, I didn't understand the paper at all, only the abstract. There was lots of physics and long the take-home message was that the Earth is spinning really slowly at the moment, and it used to spin really, really rather fast. And that's because of tidal friction and the distance between the Earth and the Moon. And that was common knowledge. I'd never thought about this, and I thought, wow, this is amazing, because what about circadian clocks? Because circadian clocks, we know that dinosaurs, they probably had a circadian cycle. Certainly birds have got fantastic circadian cycle. But in the blue-green algae, the cyanobacteria, they've also got clocks. And so they must have evolved at a time when the Earth was spinning much more rapidly, and so clocks have actually evolved over evolutionary time to be slower. And we wrote a piece about this, and lots of people in the chronobiological world were really rather interested by it. So and since then there's been work done on clock genes in the cyanobacteria. It's a different machine, but it's still a biological clock. Okay, and since we know that life evolved in the oceans, I'd just like to leave you with a couple of slides which hopefully will inform you that the complexity of clocks is much more diverse in the marine environment. And a real champion of this was Ernest Naylor. He's retired now, but he said that there was sort of two very separate clocks. It's a circadian clock and a tidal clock to be able to anticipate the tide. And more recently, there's been a lot of work done on a polychaete called Platinureus tomarilli, and a colleague of mine, Tesma Reibel at the University of Vienna, they've done some really, really smart work to try and understand what the lunar clock of these organisms is. They spawn on a lunar cycle, and even if they knock out the circadian clock of these worms, because they have a circadian activity pattern, the lunar clock is still actually functioning. And this is an isopod, and it lives on these beaches around the coast. This is a colleague of mine, David Wilcoxon, and he's been looking at the clocks of these organisms. They have a tidal swimming pattern, so as the tide comes in, they come out of the sand, they swim around, and they feed. They've got these great big beautiful eyes, so they're completely tidal, but they have these chromatophores on their back, which become darker and lighter, and that has a circadian cycle. So using pharmacological agents such as casein kinase, they've been knocking out the circadian clock. So they've been making these things arrhythmic, and seeing, even with the circadian clock, which is not working anymore, whether or not the tidal clock works. And sure enough, it seems to work. So the take-home message is that marine organisms have got really much more complex clocks than we ever thought was possible, and that they are separate from the circadian clock that we know so much about. So I think apologies that may have gone on a little bit longer than I expected. There's a lot of collaborators that I've worked with over the years, and like with all things, there's tremendous input from different labs to build up these stories, without whom none of it would have been possible, and of course all the funding agencies and institutes. Thank you very much.
Coming back to the plankton migrations, whether this happens up south and whether the lunar cycle affects when it's not the polar night or anywhere else. When the lunar cycle, does it add up to any of the migrations when we're not high?
You mean when it's not at high latitude?
Yeah.
Yes, it does. It does. It does influences, and it has been shown at fairly southern latitudes. So in the Canaries, and in places like Hawaii, they've shown the influence of moonlight on the extent of the migrations, and so something similar occurs, but it doesn't occur as dramatically as during the polar night, and that's presumably because there's a lot of sunlight as well. But absolutely, yes, it certainly does.
All right, you mentioned you were working on the big project with two species, the activity and during the night and the migrations. And my question is that whether you have the correlation between the activity and respiratory data, you measure the respiration and the behavior.
Yeah.
What is the correlation between these two?
Yeah. So I didn't show these data. We did population respirations over time, and we found that there was higher respiration rates during the night time. But those data were quite noisy because it's the whole population. So those experiments were done in bottles with lots of individuals. And so now we're actually doing trying to do those individual respirations over the day-night cycle, which I think will improve our resolution quite a lot. We didn't have that capacity at first, but yeah, the indication, as far as I can remember, was that respiration was higher during the night time than during the daytime, presumably because they are actively foraging during the night time versus the daytime.
In case you're shy, we may stop the public questions and answers session, and we still have him around for some maybe 10-15 minutes. Okay, thank you very much. Thank you.